good evening, everybody, depending on where you are, and welcome to this first bioinformatics tutorial put on by the Bioinformatics Core. I'm Larsson Omberg, and I work at Sage Bionetworks, who's one of the participating centers in the Bioinformatics Core. And today, I'm going to be talking about the first in a series of tutorials, and we're going to be about how to access the PCBC data and how to explore the data that is currently stored in Synapse. In this series, a little bit of background about it first, as a series where we start out very basic about accessing data, but go on to explore what kind of analysis you can do with bioinformatics. And we hope to be covering such topics as data exploration and quality control to see what kind of information is available in data that they might have, or data in the PCBC, and how you correct for quality issues. We would cover differential expression. We would like to cover eventually network analysis and pathway analysis, so looking at what signals are available and how does that look as when you project it onto a pathway. And then throughout the whole series, we'll be covering data visualization as well. We'll have specific sessions on covering data visualization. So today I'm going to be talking about expression analysis mostly, and exploring and fetching that data. It's important to note that the data that's available from this characterization core and what's available in the data portal, not only expression data, but also methylation data and microRNA expression data. As we go through, we'll cover looking at all of these different data types. But for the first one, we're going to talk about the idea of accessing data and how to explore it. And to do, we're going to move over to Synapse, which is the tool that we at Sage Bionetworks have developed for working collaboratively in consortia. And the Synapse webpage, you're greeted with basically a login screen with a little bit of information how you get started. But if you haven't already created an account, the first thing you'll need to do is register for Synapse. It's relatively easy. You need to choose a username, input an email address, and your name. And once you have done this, you'll end up receiving an email verification, which is really important that you press the link on that is provided in that email. Otherwise, your registration will not be complete. And if your registration is not complete, you won't be using your account in Synapse. Once you've created an account, you can go to the login page of Synapse, and then you can type in your username, password that you've chosen when your account. And once you sign in, you'll be greeted by your space in Synapse. And so depending on how long you've been using and how much you've been using Synapse, depends on how much you'll have. So for example, this is the way it looks for when I log in. So I have my projects, which are the projects that I'm currently involved in. I have other people's projects and files, etc., that I found interested in, so I've added favorites to those. And then I also have teams that I'm involved in. So these are collaborations that I'm involved in. For me, for example, you can see there's a lot of things about stem cells, a lot of things related to cancer, which are the two fields that I work in. So what we're interested in is the progenitor cell biology consortium. And when you come here, everything will be blank, and it's important that you get access to this project, because this project is actually private and only visible to people who participate in the PCBC. And you'll have to do, once you've created an account, you'll have to reach out to me, and I will add you as a participant in the team for the PCBC. And options for this is available on the main page for the PCBC. If you look at bioinformatics and genomics tools, you can click Synapse, and there's the actual information. And I agree, a little bit clunky, and we're working on making this registration process a little simpler. But in the meantime, you'll have to do that, and once you have done that, you can go to the progenitor cell main page, and to make sure that it pops up in your main, and you can click the little favorite button right there, and once you log into Synapse, it will be in the front page for you to see your file. And what I do today is go over a little bit about the different parts that we provide. But before that, I'd like to highlight the fact that if you run into trouble and you need help, there's a forum which is staffed by people both at Sage and then also people from the Bioinformatics Core if you're asking for specific questions on PCBC, and ask questions that relate to that. So for example, we can see what other questions have been asked by other people, and it turns out there's only one question currently that has been asked. But if we go back to the front page, the question what about this data? And I continue. And of course, if this is a question that's already similar to something that might have been asked, it will show you that you can look for those. But if it's really a new question, you can in details about your question. And if you want to address it specifically to the PCB, it helps you choose to say that's related to the PCBC. This question will be directly emailed to you and other participants, whilst you do a general question will ask it to anyone who's participating in Synapse. It will eventually get answered, but it will get quicker to answer if you choose to use the PCBC. I'm going to close that out and go back to the main page of the PCBC. In the project, we have a wiki that describes the content, and then also explore all the files that have been contributed to the project. But it's generally easier just to use what we provided in the wiki, and we divided it up into just certain categories that might be useful: extracting your signal in the self-characterization data, downloading data, exploring how we process the data, and then as results that have come in, and it links out to other tools that might be useful, such as enrichment analysis from Cincinnati Children's Hospital. And I'm going to start at the top level, which is actually if you have a signal that you're interested in exploring, or you have a set of genes that you want to see how they behave in the people data, provide a tool for that. And then I'll move on to more detail, download the data, and I'll explore the data locally. So let's start by looking at exploring the signal. We have provided basically a viewer into the expression data. And so if I pull up here a little bit, what this is, it's a hierarchical clustering that's being done on all of the expression data that's currently in Synapse. So when data is added, this will be updated as well. The way it works is that you can input the set of genes that you're interested in exploring, or you can choose specific pathways that you're interested in. Certainly, it's using a list of genes, and it's displaying those genes here, and then the samples across here. And they're highlighted currently by the differentiation state. So that embryoid body, ectoderm, mesoderm, other actually tissues and stem cells. And you can see that stem cells cluster by themselves and the different differentiation states differentiate across. And that's because the list of genes here are genes that are very relevant for the differentiation states that were chosen. We can also choose to look at specific pathways. So we can choose a pathway, and if we go bottom, we'll choose the Wnt pathway. What it's doing now is pulling out all those genes and the expressions that are part of the Wnt pathway and showing how those cluster across the samples. Or we have also pre-computed differentiations between different states or cell origins, also differentiation based on states, and choose those gene lists. So, for example, you can choose those that are both in ectoderm and those that are derived from blastocysts, so embryonic stem cells, versus those derived from amniotic fluid. And you can then also further filter those down by specific pathways. So this gene list is enriched for the following PEG pathways, and if I click on a specific pathway, it filters down to those genes that are part of that pathway and also enriched, for example. Let's back to the gene list, which is you're coming at this where you have a data set, you've already found a set of genes that are interesting, and you want to see how those genes behave in this data set. So we can place these, for example, and we just pick a few genes that SOX2, MYC, and KLF4, and maybe NANOG. So picking 4 genes that are used to differentiate stem cells, for example, and we can see those genes now behave across all of the samples. This is a lot of samples. We might not want to see all the different differentiation states. So what we'll do is we'll fill them down and only look at those cells that are part of the embryoid body and the stem cell state, basically. So differentiated and those that are slightly differentiated. We basically see a new string, and we can see that basically the EBs cluster well together with the stem cells, and see how the expression changes across the samples. Now, you see this outright outlier here, so this might be a quality issue where one of the samples is measuring the MYC, for example. And then it's important that you can also change what you're filtering by. So, for example, we can add gender, So now it'll tell you what the sample of the sample where they're derived from, and you can see that basically the gender distributes evenly. So this is not a signal that's driven gender. We can line type, so it'll tell us whether it's a human embryonic stem cell or an iPS derived stem cell, and also pick cell origin. And you can see basically this is very robust for the EB versus stem cell. And the important part, though, is that I'm just picking something randomly out of the air, basically. But the point, you can take your set of genes and see how they behave in this. If you found something that could be interesting, you can explore it further. So you can basically see what the genes, some more information about the genes. And you can search within those, that gene list as well. So if you choose a pathway or significant enriched gene list, you can find out what genes are in it. And then importantly, you can just download this data. So what this does is it fetches those, in this case, 4 genes, and gets the expression data across all the samples for these 4 genes. And you can click on an enrichment analysis, which take these 4 genes and pass them over to the top gene program, where you can look at how well those genes are enriched for different classifications. So go back to the top here, you can also see where does, we'll go to that heat map again, so where does data come from? That's the important part. And so we have information here that this data is actually derived from something that's derived by a SYN ID, and this is how it is part of Synapse. So we can actually click on that, and we see where this data comes from. And this is all the expression data, all the samples that are part of the cell characterization core. And then after you have a little bit of information about it, you can see the size of the file. It has the ID that's really important. We're going to use that later to actually fetch data. And then it has information about the history, so we can see currently there's only 1 version of this file. And then you can see a little bit of what the file looks like. Where were this file derived from? So what was done to achieve these expression calls? Well, for that we have a provenance graph, and if we could spend, we can see this is something that used concatenation, which script, and the files in this folder. So basically this file was used by combining the files in this folder. So we could go over to that folder and see what these files look like and what they contain. And what you'll notice is that the page, it's a long list of each of the samples with their FDM, which is a measurement of expression of genes and isoforms. So if we click on one of these files, again, we see we can download this file. So this would be the expression call for just 1 sample instead of the kind of all. But then again, more importantly, we have the provenance of it. And so what this is that the file was generated by running cufflinks, and we can see that it's an older version of cufflinks. So maybe it's time to run this data with the newest version of cufflinks. It used M file as input, and it's a gene model that's available from Sanger. And then you see the script that was run, which tells you the pointers that was run to call the externalities using cufflinks. The BAM file, you can find out where that was derived from. Well, that was actually run using PubPad. Again, the parameters are available in the script and was used using the FastView file. So you can download this data at various stages of how raw you want it, depending on what kind of analysis you want to do. But in the easiest is actually to go back to the real data, and so we'll go to the wiki and we'll go to downloading. Actually, the file for the expression calls. Similarly, we have files for the mRNA and methylation and information about the version, who uploaded it, and a short little description. And that's on top of that, we also have metadata and the raw sequencing data that I showed you as we tracked in the provenance graph. But let's go back to this expression call data, and let's assume I want to download this for further analysis. I can click on it right there and starts downloading it. It's a 53 MB file. Most of the time when you're actually fetching data and analyze it, you actually don't want to just pull down the file. You actually might want to work with it directly in a programmatic way. And we're jumping very far here from interacting with it on the website to a programmatic client. And so what I'm going to show here is using IPython, which is very similar to it's a way of it to write prompts and interact and do computation on it. And it sounds advanced. Think of it as just as basically a super advanced calculator. I can type in formula and it can execute that formula. make it a little bigger so it's easier to see. And what I'm going to do is I have imported some of the things that I will need. And what I do is I'm going to fetch the file that we talked about earlier. So I'm going to do syn get, and the ID of that thing was syn 247799. So that was the file we were currently looking at. And what this does is it goes to Synapse, fetches the file, and downloads it to the local computer. Now it's going to be very fast as I do this, and it's very fast because I've already fetched this file. What it does is it goes to Synapse and asks, is there a newer version of this file? If there's not a newer version, use the one that I've already fetched. And then we see if we look at this, this file has some information. We can see where it's stored. But more importantly, we probably actually want to read it here so we can do some analysis on it. And so I'm going to use pandas, and I'm going to read in. We're going to read the file that's stored in the path, and it's a time-limited file. I know that because it ends in TB, so I know the separator is equal to T. Know that the genes are stored in column 1, so I use that as an index of this. And to store this in a variable I call EXPR, short for expression. And if I run this, I'm now reading that file into memory. I can look at what this file looks like by looking at the head of this file. And notice this is exactly the same kind of information that we saw on the preview on the website. So basically, we have gene names along the rows, and then we have sample names along the columns. And we also have some information about the Hugo as the name, and we have the ensemble name and the position of the gene that they're located, and then expression values. And if it's about this, we can actually add information about this as well. So we can actually let's assume that we want to see what does this gene each one look like. So we can extract that gene EP1 and we extract the expression value, and then we're going to plot a histogram of that. And what we see here is that we end up with a histogram. This is the expression value of those genes across that. So the kind of thing that we'll be doing in more advanced sessions, we'll be processing the data and looking how the genes look. And we can do a comparative analysis, very simple here. We can actually look at this, and then let's compare it to nanog. And I just need to transpose this to make it work properly. And do this. I have the distribution of the expression of 2 different genes. And just as I've fetched the data, I have looked at specific ones in the data, and even move further than that. I do, if we look at a matrix, it's actually very large. It's about 57,000 different probes, and that's a lot. So let's filter it down to something more manageable. So I'm going to do the standard deviation of each gene, and we're going to keep those that are above 10. So I'm keeping the most variable genes in this. And what I can do is I can extract those, extract the expression values for those, and I'm going to store that in a variable small. And then we'll just look at how many samples we're left with when we do that. The reason I'm decreasing it's just so it's a little bit more convenient to compute on. Now we have something that's about 7,000 genes versus 170 samples, and now I can actually do an SE analysis, which is basically a PA analysis on it. And so I can now look at the small variables. And this is a script that I have that very quickly does a PA to quickly visualize what the data looks like. So we're getting a little bit about looking for quality in the data, etc. And we'll do more of this in the future analysis. So it's taking a little while because it's on a laptop. And what we see here, it generates a bunch of things about the quality of data. So this is basically the PA in summary form, and then the more traditional PC plot, so we can see what the distribution of the data is. And it'd be a lot more interesting if be color-coded these by some information, such as what maybe the origin or the differentiation method, etc. But this is just showing how easy it is to test the data and then do a quick analysis, look at quality control, and then equally we could have done something for looking for differential expression. If we go back, I'm going to go back to the web page just to summarize. We go to the front page. What we're providing here is we can provide a way for quickly exploring the expression data. We'll soon going to be adding the same kind of exploration using this heat map viewer for mRNA and methylation data as well. It'll allow you to download the data. You can just download from a website, or you can interact with it directly from a programmatic client. And this is kind of the things that we'll show for in future bioinformatics tutorials. I think I'm